talk I would like to explain a little bit more what we expect from regulatory T cells in amelioration of graft versus host disease, and ask the question whether this platform can allow the cure rather than the control of HIV infection, and whether this is feasible. Now, it's well known that the concept of allogeneic stem cell transplantation of the cells resistant to HIV has been used successfully in the case of so-called Berlin patient. This is the case when Timothy Ray Brown, who suffered both from leukemia and HIV infection, has been cured after transplantation of hematopoietic stem cells conferring delta 32 mutation in CCR5 gene, and this allowed to cure both leukemia and HIV infection in one step. So we believe that it is possible to develop regimen to improve and modulate the function of regulatory T cells, Tregs, which favors suppressing graft versus host disease, GVHD, while retaining the benefits of graft versus leukemia and graft versus viral reservoir effect. So in order to achieve that, we intend to create database of bone marrow donors as well as umbilical cord blood cells, which are homozygous for CCR5 delta 32 mutation and therefore resistant to HIV infection, R5 strain HIV infection. We know that under these circumstances, the biggest threat of, after bone marrow transplantation, the biggest threat of graft versus host disease could be potentially fought and ameliorated based on detailed knowledge of cyclic adenosine monophosphate, mediated suppression of alloreactive T cones, using Tregs harboring delta 32 mutation. And we will strive to improve Treg immunosuppression using HIV glycoprotein GP120 to ameliorate GVHD. And I will explain in a minute how. And third but not last, for clinical application, we believe would be important to get insight for treatment of umbilical cord blood cells heterozygous for CCR5 delta 32 mutation, to use prostaglandin E2 mediated mechanism, which can facilitate both allogeneic transplantation. There is a clinical trial performed in this regard at Dana-Farber in Boston. And based on our own data, PG2 has also capacity to downregulate residual CCR5 expression and therefore make these cells more resistant towards HIV. And key advantage of this process would be the less stringent requirement for HLA matching and better engraftment of the cells resistant to HIV. Now, it should be emphasized that Berlin patient been a major success in this regard and really allowed cure of both conditions in one procedure. However, exactly the same strategy used in SM, in the case of SM patient, been plain disaster leading to death. And prior to that, the SM patient had been also viremic, and showed that the CXCR4 tropic HIV can again re-emerge despite that HSC transplantation was performed from CCR5 delta 32 donor, because these cells are still susceptible to X4 tropic HIV. So we believe that in order to advance this field, it's vital to search for novel determinants of HIV susceptibility based on in-depth comparison between Berlin and SN patients using massive parallel gene expression and new generation sequencing. And this is based on the fact that HIV is affected more than 2,400 protein coding genes, which represent nearly 10% of all human genes in human genome. We also need to look very closely for transcriptomes of primary CD4 and CD8 T cells, because there is an evidence for network of enriched pathways associated with HIV disease. So single genetic variant such as CCR5 delta 32 mutation may have a very different impact depending on the other genetic variants that exist in the genome, these so-called modified genes. And of course, environmental factors or combination of those. Now, we reviewed this in a review or paper last year and this year, and in principle, intracellular immunization with gene-modified hematopoietic stem cells, as was proposed by Hans-Peter Kien at Fred Hutchinson Cancer Research Center, is based on the very simple idea: you collect a few hematopoietic stem cells, they are treated, such as prostaglandin-E2 treatment, or selected such that they confer naturally occurring mutation conferring resistance towards HIV, like CCR5 delta 32. They are expanded, infused, and a new immune system where HIV-resistant blood cells are generated inside in the patient is established, and both T cells and monocyte and macrophages are resistant towards HIV infection. So in principle, it's very simple. There are, unfortunately, caveats, as I already mentioned. We know that HIV needs for its entry interaction with through GP120, and which is on the spike here, and CD4 receptor, where co-receptors such as CCR5 or CXCR4 are required. Now, for initial infection, CCR5 co-receptor is critical, and therefore this CCR5 delta 32 mutation, which prevents CCR5 expression, can prevent also effective infection by R5 strain of HIV. Now, what is less well appreciated is the fact that the GP120 interaction with CD4 has also intracellular consequences, signal transduction consequences. So GP120 or GP120 derived biologicals, then when they see regulatory T cells, which is also CD4 positive T cells, but with CD25 receptor, IL-2 receptor expressed, co-expressed in the same time, will trigger the signal transduction pathway, which is leading to activation of adenylyl cyclase and a profound increase of intracellular concentration of cyclic AMP, opening up gap junction, which in contact-dependent fashion is responsible for interaction with effector CD4 effector T cell, and literally injection of this endogenous cyclic AMP into this target suppressed effector T cells, which has dual effect. One is proximal, leading to obliteration of TCR signaling and energy, and the other is nuclear and leads to induction of inducible cyclic AMP earlier repressor, which stands for ICER, acronym of this is ISA, and this is a very powerful transcriptional repressor which is involved in transcriptional attenuation of interleukin-2 expression. Now, interleukin-2 expression is very important for activity of regulatory T cells because they express very high levels of interleukin-2 receptor. Now, this has dual effect. One is pathology during HIV infection, leading to reduced HIV-specific immune response. And what we would like to achieve is to hijack this pathological effect for therapeutic effect, mainly for reduced tissue rejection or reduced autoimmunity. Of course, we will focus more on reduced tissue rejection because we know that during bone marrow transplantation, 50% of patients suffers by graft versus host disease. And if we are supposed to translate this into the clinic, we have to deal with graft versus host disease in efficient way, more efficient way. Now, we already know more about CMP-mediated transcriptional attenuation of IL-2 and Treg role in this mechanism. During the last several years, we learned to our surprise that ISA, which is in blue here, is open activation with CD3, CD28 ousted to the cytosol, and in the same time, nuclear factor of activated T cells is translocated to the nucleus and allows a vigorous IL-2 expression in these cells, right? So here is overlay, ISA is in the cytosol, inhibitor is in the cytosol, activator is in the nucleus. However, if intracellular levels of cyclic AMP are elevated using pharmacological agonists such as forskolin, which directly activates aryl cyclase, then both ISA and F18 colocalize in the nucleus, and this is reflected by transcriptional attenuation of IL-2 expression in these effector CD4 effector cells. Interestingly, if you isolate negatively conventional versus regulatory T cells and then measure intracellular CMP levels, you can see that there is nearly 20-fold difference between them. So Tregs have naturally very high levels of intracellular cyclic AMP, and we believe that at least part of it is involved in interesting observations. This is confocal analysis of Treg cells, and as you can see, no matter what you do with them, whether you prime them with CD3, CD28, or co-treat them with forskolin, they remain the same. ISA in the nucleus and NF18 in the cytosol, and that is a bit surprising because, let's say, Ajahn Rao proposed that NF18 and FOXP3 in the nucleus of Tregs are the most important complex for T cell tolerance. And since NF18 does not even translocate to the nucleus, chances are that this is not really taking the place, and the whole mechanism of tolerance in this regard is dictated differently. So just to summarize, CMP-mediated mechanism of Treg suppression, of suppressed or T cell in contact-dependent fashion, we propose that, or there is a sufficient accumulating evidence suggesting that FOXP3 can inhibit phosphodiesterase 3b, which prevents then conversion of CMP or degradation of CMP. And in the same time, through MIR 142-3b, which inhibits aryl cyclase, in fact, aryl cyclase is activated and CMP formed to 10-fold, nearly 50-fold difference between Tregs as far as CMP is concerned and suppressed T cell. And the contact-dependent nature of this process allows gap junction communication to transfer CMP into the suppressed T cell, induce ISA, inhibit IL-2. And this is based on very orchestrated nature of yin and yang dance of ISA and F18 switching position between cytosol and nucleus. Kjetil Tasken also has shown that in Tregs, in the presence of elevated level CMP, this gap junction, which consists of CX43 monomers, can get phosphorylated, and they are opened up and allow this injection of CMP into the neighboring suppressed T cells. However, Sitkovsky and colleagues also shown that there is a contact-independent mechanism which is based on hypoxia adenosinergic signaling. In principle, there are CD39 and CD73 ectoenzymes which can convert ATP to ADP, AMP until till adenosine is cleaved off, and then adenosine can interact with adenosine 2 receptor, and elevated intracellular levels of cyclic AMP induce ICER and form this, presumably this inhibitory complex, which is involved in transcription attenuation of IL-2. So basically, this operates through the mechanism where, in the context of IL-2 promoter, CD28 responsive element is occupied by CREB, open activations, phosphorylated recruits CREB-binding protein, and NF-8 is translocated into the nucleus and forms a vigorous complex which is responsible for IL-2 expression. And while in the presence of Tregs or elevated levels of cyclic AMP, ICER is induced, outcompetes ICER for CREB for binding, because this induction is robust, 20-fold, 50-fold in comparison with CREB, which is obvious, ubiquitously expressing relatively low levels, and open activation. NF-8 ICER complex could be formed, and this yields a suppressed or anergic phenotype in T cells. Lin Chen and myself, we collaborated, and we are now in the process of crystallization of this complex. This is just the first glimpse of the model, where ICER as a dimer binds to DNA. This is CD28 responsive element motif, and this dimer is ICER, and interact with NF-8 interface in a very similar fashion as NF-8 Fossa and June does interact. This has been already crystallized. The only difference is that this is activatory complex, while NF-8 ICER is inhibitory complex, which we believe would be central for tolerance. And if one compares the residues, amino acid residues in C-June, CMAF, and ICER, then it seems that ICER have perhaps even higher affinity in this protein interface than these other members of the family. So one could conclude this part that CMP underpins suppression by regulatory T cells, and that this communication is achieved through gap junction intercellular communication, and particularly for suppression of antigen-presenting cells or dendritic cells, also expression of CTLA4, which is considerably expressed on Tregs, and B7, which is expressed on DCs or antigen-presenting cells, could also allow induction of ICER and be instrumental in this process. So one can end up with model like this, where Tregs inhibit suppressed T cells in the presence of APCs, and instead of triggering immune response, this complex is, or this three cellular interaction is involved in induction of tolerance. Now I will switch the gears a little bit, and will refer about prostaglandin-modulated umbilical cord blood hematopoietic stem cell transplantation. That's a clinical trial one at Dana-Farber, published a few years ago, where pulse treatment of umbilical cord blood cells with dimethyl PG2, which is a more stable agonist or analog of prostaglandin E2, is safe and may lead to accelerated UCD engraftment and preferential chimerism. And we also know, based on work of Kjetil Tasken that PG2 has capacity to induce FOXP3 in adaptive Tregs and in such way contribute to suppression of effector T cells using mechanism which is outlined in the section B. And for lack of the time, I will go directly for our original observation, which we have done years ago at National Cancer Institute, where we have noticed at the time using RNase protection that PG2 indeed can attenuate CCR5 expression after 1 hour, so it's relatively quick, and this is accompanied by robust ISA induction. So from that point of view, there are several approaches to modify hematopoietic stem cells for HIV resistance. Of course, one is transient CCR5 knockdown, which would be achieved by using dimethyl PG2, as was suggested. There is a clinical trial ongoing right now, which is using permanent CCR5 knockout strategy, using CRISPR, Cas9, Zinfinger strands, etc. So genome editing approaches seems to be very promising in this regard, because CCR5 does not seem to be essential for any developmental function. And last but not least, integrating the delivery vectors would be an option to achieve the HIV resistant cells. I would like to briefly mention our pilot studies regarding CCR5 genotyping, which we have done in Prague, Czech Republic. We learned that we can detect very high frequency, at least to my perception, of heterozygous donors or individuals, which here is allele with delta 32 mutation, around 20% in Prague. For homozygous individuals, we found numbers rather limited, around 1–2%. Indeed, when we sequence these PCR products, we found that this delta 32 mutation is there exactly as Ned Landau and Mary Carrington described back in 1996. Now, geographic spread of CCR5 delta 32 allele is clearly well represented in Norway, Finland, northern part of Germany, Ukraine, and also central part of Europe. I think that allows us to search for this mutation. It's kind of gift from Mother Nature, since in peripheral CD4 T cells, frequency of Tregs, regulatory T cell, really does not depend on genotype. And these cells can be effectively expanded after CD3, CD28 activation in the presence of exogenously added recombinant IL-2 from 7% to 80% within a week. So this is a modality which really could be clinically used, and hopefully on our improved knowledge of cyclic AMP-mediated suppression and GP120 treatment could be used for amelioration of graft versus host disease. Or alternative approach, of course, would be to utilize dimethyl PG2 for transcriptional attenuation of residual CCR5 expression and making these cells resistant to HIV. So I think I will stop here. Prior to doing that, I would like to acknowledge my collaborators in Prague, Petr Kobylka, Julie Chalupníková, Kateřina Pavelcová, Pavel Jindra in Pilsen, Edgar Schmidt, Hans-Jörg Schild, Helmut Jonuleit, Christian Becker at Uni Mainz in Germany, Joachim Haube, Jan van Bonsen at Uni Hamburg in Germany, Kjetil Tasken at University of Oslo in Norway, Hans-Peter Kern at Fred Hutchinson Cancer Research Center in Seattle, US, and last but not least, Gero Hita who is the first who be able to cure HIV by bone marrow transplant of the cells resistant to HIV, who is now at Salex in Dresden, Germany. Thank you very much for your attention.
Well, the questions, please, from the floor.
Thanks for your presentation. It is very good. And I want to ask if this treatment can be given to attendants with HIV, no HIV disease patients, to persons left with HIV patients. Can we do it?
So yeah, I just will try to repeat the question. So the question is, you are asking whether this treatment
Normal people, not children. Yes,
can be provided to HIV patients who are without malignancy.
Yes.
Well, it's a little bit complicated at the moment. I think at the moment this is viewed as unethical. And the reason is that after bone marrow transplantation, the incidence of graft versus host disease is around 50%. And until these numbers are not more favorable, means if we are successful with GP120 and we can achieve, let's say, 80–90% amelioration of graft versus host disease, then I believe that we will get to the point where it would be ethical to treat HIV patients with bone marrow transplant from the donor who is resistant towards HIV. Yes.
So this is only for HIV-1, right? Not for HIV-2, or does the HIV-1 mutations affect the effectiveness of the transplantation?
That's correct. At the moment, we even have to say that this is only effective towards HIV-1 strain, R5 HIV-1 strain, CCR5 HIV-1 strain, because later in infection, usually after several years, there is a shift towards CXCR4, so-called X4 HIV-1 strain, and that unfortunately CCR5 delta 32 mutation, which leads to limitation or to decrease or let's say even absence of CCR5 expression as a necessary co-receptor is not sufficient to protect against X4 HIV strain infection. So even so, we know from the case of Berlin patient that there is indeed potential to cure leukemic patient with HIV and get rid of both leukemia and HIV infection. In the case of SM patient, this approach has not been successful, and there has been a shift towards X4 strain. So we need to understand more both about HIV tropism, but also about the genes, these so-called modifier genes, which are probably different in both cases, Berlin versus SM patients. So the feasibility is clearly there. We should be able to achieve this, but we unfortunately need to learn more how to do it.